I am Dr. Khokon K Dutta. I have a Christian name, Dr. David Brown. I'm currently working as assistant professor in the Department of Biochemistry and Molecular Biology in Bangabandhu Sheikh Mujibur Rahman Science and Technology University in Gopalganj, Bangladesh. The title of my discussion is First Ever Experiment Internationally that relates the roles of Txnip or TBP-2 or VDUP1 with non-cancerous cellular proliferation and regeneration. Khokon K Dutta et al. From the School of Medicine, Kyoto University, Japan, for the first time reported that TBP-2 or Txnip or VDUP1 works as a tumor suppressor in vivo. We used oxidative stress induced renal cell carcinoma of rats as the experimental model. The report was published in the pathology section of Nature Laboratory Investigation in 2005. We found that the reduced expression or the absence of TBP-2 or Txnip or VDUP1 is essential for cancerous growth and proliferation in rat renal tubular cell carcinoma. Subsequently, TBP-2 or Txnip or VDUP1 has been reported as a tumor suppressor for many other cancer types. We also checked the roles of TBP-2 or Txnip or VDUP1 for the regenerative proliferation and growth of the non-cancerous cells in the rat kidney. This was the first attempt internationally to find out the importances of TBP-2 or Txnip or VDUP1 with regenerative processes in any tissues or organs. In our experiment, we injected an iron-chelate ferric natriuretic FENTA intraperitoneally in a repeated manner for 3 weeks. We collected kidney tissues for Western blot analysis and immunohistochemical experiments. We found a gradual decrease for TBP-2 proline with time. At the same time, we found an increase in proliferating cell nuclear antigen PCNA with time. It would be nice to mention here that PCNA is an indicator for new cell division. FENTA administration induces cells and tissue damage in the kidney of rats. After a certain time, cellular regeneration starts, which explains the increase of PCNA proteins. In another experiment, we used serial sections of FENTA-treated kidney tissues to localize both TBP-2 and PCNA on different sections for comparisons. We found something surprising, where PCNA positive cells were negative or found with reduced level of TBP-2 protein. PCNA and TBP-2 proteins were inversely correlated. Reduction under a certain threshold for TBP-2 was found to be needed for a cell to divide and proliferate in a regenerative mode. Our experiments suggested that reduced level of TBP-2 is an essential signal for regenerative growth in damaged tissues. In today's world, diabetes is a big problem for all the countries. About 463 million adults are suffering from diabetes presently. Many others are in the pre-diabetes condition. People with diabetes are also suffering badly in the current pandemic with coronavirus 2019 or COVID 19. It is also known today that TBP-2 or Txnip or VDUP1 is the central player of the detrimental effects in diabetes and diabetes related complications in different organs like pancreas, kidneys, heart, eyes, brain, skin, and others. TBP2 or Txnip or VDUP1 inhibitors are seem to be able to tackle both the types of diabetes, type 1 and type 2 diabetes, very effectively. Mitochondrial TBP2 or Txnip or VDUP1 is the key regulator of enhanced ROS production and inflammasome activation and inflammation. TBP2 or Txnip or VDUP1 inhibitors are able to stop the reactive oxygen species and inflammation mediated insulin producing beta cell death in the pancreas. Scientists already proved that deletion of TBP2 or Txnip or VDUP1 in the pancreas induce an increase in beta cell mass in the pancreas by stimulating regenerative growth. So, a good inhibitor for TBP2 or Txnip or VDUP1 hold the promise to tackle all types of diabetes and to reverse the damages in the pancreas to back the normal number and size of the insulin producing beta cells. Finally, I would like to wish a very happy, prosperous, and dignified life for myself and for you everybody in the light of the true values for freedom. Thank you so much.